Oh. Okay, thank you all for coming to the first BGSA seminar series. Just to start, I will start with the land acknowledgement. We would like to respectfully acknowledge the island of Newfoundland as the ancestral homeland of the Mi'kmaq and Beothuk. We also recognize the Inuit of Nunatsiavut and Nunatkavet and the Innu of Nitassinan and their ancestors as the original people of Labrador. We strive for respectful and equitable partnerships with all the people of this province as we work towards true reconciliation and honor this beautiful land together. So it is my pleasure to introduce Dr./Father John McCarthy, who is a Jesuit priest and scientist. He was ordained in 1994, and along with his philosophical and theological studies, he received a bachelor's in forest tree biology from the University of New Brunswick, as well as a master's in soil science from the University of Florida and a doctorate in boreal forest ecology from the University of British Columbia. Dr. McCarthy is a lichen specialist and has collaborated in naming of several species of lichen new to science, such as Acarospora McCarthyi. Yes.
I didn't name that one though.
He speaks on subjects such as Christian faith and ecology, as well as faith and science, and publishes on lichenology and ecological theology and spirituality. He currently serves as superior of the Great Lakes North Jesuit community in Northern Ontario. He works on the lichen biodiversity of Newfoundland and Labrador as a visiting scholar in the Department of Biology, Laurentian University in Sudbury, Ontario. Without further ado, please take it away.
Well, thank you, Deepal. Thank you very much for your generous invitation. I wrote it, of course, but it's good. So thank you very much for being here this afternoon. I certainly appreciate your presence, and also for those who are here online. And I understand that some of you are here for, you may get extra credit if you're here, but that's okay too. So thank you for being here. I'm originally from St. John's, actually, from St. John's, Newfoundland. I was here at Memorial in 1977 to 1974 to 1977 was the pre-forestry program. And then we went to the University of New Brunswick. So I'm kind of dating myself somewhat. But it's good to be back in this brand new building. So I'd like to begin with just with a quote from Charles Darwin. If you read Charles Darwin's On the Origin of Species, the 1859 or 1860, have it next, yeah, 1860 edition. If you go to the last paragraph of Charles Darwin's book, the best way to understand a book is to read the introduction, then read the last chapter. Everything else in the middle is filler, the argument for the book. Anyway, so the last thing, it is interesting to contemplate, he says, an entangled bank, clothed with many plants of many kinds, with birds singing on the bushes, with various insects flitting about, and with worms crawling through the deep earth, the damp earth. And to reflect that these elaborately constructed forms, so different from each other and dependent on each other in so complex a manner, have all been produced by laws acting around us. ... There is grandeur, he says, in this view of life. From so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Ooh. So endless forms, most beautiful, have been and are being evolved. This is a wonderful way, I think, to look at least from my angle, look at lichens. So I'd like to share with you this afternoon my engagement with, my love of things most beautiful and most wonderful, the lichen biodiversity of Newfoundland and Labrador. Just a bit of a roadmap, just a little bit on the biology of lichens, what's a lichen, a bit on the history. It's always good to know where we come from in any discipline, any discipline at all of knowledge. It's good to know some of our history. And then we get into Newfoundland. Why is Newfoundland and Labrador a haven for lichens? And it really is. It's really an important biodiversity center, really, for many species across Canada. And then finally, the last two sections, or what's in a name, describing new species. a bit on my work on lichen taxonomy and some of the work that myself and colleagues have been doing. And finally, a little bit of history of Newfoundland and Labrador lichenology and the future of lichenology in Newfoundland and Labrador. And the future is found in this room as well, excluding myself. And just a final question, why study lichens? Why do anything? You know, why are you in biology? I assume many of you are. Why do you study what you do? And why do you enter the science of biology? So first of all, what's a lichen? It's maybe a bit small, but to give you some idea that for a long time, we didn't know what lichens were. We didn't know where to place them, especially Linnaeus in the 18th century. Finally, we came to realize that lichens are indeed fungi. They're in the fungal kingdom. So if you look at the 5 different or 6 different kingdoms, from Anabacteria over to the animals on the right, and about half of the fungi, as we know, are saprophytes. In other words, all fungi depend on external carbon. They're heterotrophs. They take in carbon from an external source. So half of the fungi are saprotrophs. You know these well by mushrooms on trees and mushrooms in the soil. But the other half are what we call biotrophs. They enter into relationship with living organisms. They either kill the organism, parasitize it, or they enter into a mutualistic relationship. And so about 30% of the fungi are into a mutualistic symbiosis. And this is where you find the lichens. And of the 30% of fungi that are mutualistic, about 9% are mycorrhizae. As you know, there's no such thing as a naked root. You know, all roots of trees and shrubs and many flowers have intimately associated with them fungi in the form of mycorrhizae, fungal root. And whereas the rest, about 21% of all lichens in general, as far as we know, of all fungi are indeed lichens. So just to give you, again, just a little phylogenetic tree, fungi are more closely related to animals than they are to plants. They're closely related. You can see on the upper left, you can see, it doesn't really show here, but you can see the lichens, yeasts, molds, smuts, rusts. So they're all found within the fungal kingdom. How many fungi are there? No one really knows. There's a lot of different estimates that are developed by different techniques. But for our purposes this afternoon, maybe 2 to 3 million. But of that, say, 3 million, we only have names for 155,000. So there's a lot of work left to do. If you're interested in fungal taxonomy, you'll have a lifetime of work left. Each year, we describe about 2,000 new species. And the number of lichens, again, about 20,000, 18 to 20,000 is the figure given. All lichens, about 99%, are what we call Ascomycota. They're in the phylum Ascomycota, meaning that the spores, see 8 little spores, are contained within a sac called an ascus, and that's within the reproductive structure of the fungus. And about 40% of ascomycetes are lichenized. There's the whole lichenization process apparently evolved and came in at different times during the evolutionary history. So it's not a once-only thing. When we talk of lichenization, a very simple schema, you have this free-living algae in the environment. You'd often see it as a kind of a green fuzz, you know, on trees, on cement, on sidewalks. And they'll come in contact with fungal hyphae to create the lichenization process. And it's interesting, this process, we don't really know how it works. There's a lot of research today on what are all the molecular signals, what are the genetic bases of the whole lichenization process. So it's something that's really quite unknown, but in the end it produces, as you can see under D, it produces a lichen which is quite different than the free-living algae and free-living, say, fungal hyphae. So 1 and 1 does not equal 2. 1 and 1 equals 3 plus. It creates a whole different type of organism. We often thought, and in general, it's a mutualistic relationship between a fungus and an algae. They take a lichen to each other, and they create a lichen. That's an old, that joke's been around a long time, I think. Take one example. You know, you have the fungal tissue. This is like you kind of take a cross section of a thallus of a lichen, the fungal tissue on top and on the bottom, and the algal cells inside in some kind of extracellular arrangement, you know. And we'll take a closer look now at some. Lichenologists in general like to describe 3 different growth forms of lichens. They're not phylogenetically related, but there are kind of 3 different forms that we kind of like to, that can help us understand lichens. On the left is the so-called foliose lichens. They're kind of leafy, lettuce-like. And you can see the upper cortex, lower cortex, algal layer, and then they're connected to the ground or through the substrate tree bark by some kind of specialized hyphae, the rhizonae. In the middle, you have a crustose lichen. And the crustose lichen are often saxicolous. They're often grow on rocks, and they're embedded directly into the rock. So the fungal hyphae on the bottom will embed themselves in between the rock grains. And so if you want to sample saxicolous lichens, you have to take a piece of the rock. And then finally on the right, pendulous or fruticose lichen, 3-dimensional lichens. Here you have upper and lower cortex and the algal layer in the upper and in the lower section of the thallus. So those are like in Newfoundland, old man's beard, as you see here, all the different cladonias that you see growing around. So those are kind of 3 different general types that we have. The photobionts, most are green algae, but there are a good percentage we call the blue-green algae or cyanobacteria. And these can fix nitrogen as well. And some lichens have a combo. They don't care what they have. They can get along with green algae and blue-green algae at the same time. So let's just take an example and to run through a little, run through a bit. This is a not uncommon lichen in Newfoundland. It's called Lobaria pulmonaria, the lung lichen. And if you take, just say you take a cross section of one of the thalli, you know, just with a razor blade. And again, you can see the upper cortex. And then right underneath the cortex is the algal layer, positioned for maximum photosynthesis and maximum protection in a way. And this particular lichen has both green algae, as you can see in the below the cortex, and then a specialized blue-green algae in the specialized structure called the cephalodia, those dark green. So it has two different types of algae within this particular lichen. There's another schema just to show it in more detail. And what's often interesting now is that people are finding more and more, it's not simply a fungus and an algae, 1 or 2 algae, but they're also, even on the cortex surface, you may have specialized bacteria that are not simply ancillary, but they're intrinsic to the whole lichenization process. And you may even have lichenicolous fungi that grow within the thallus itself. So it's quite a complex thing, more complex than we thought. Toby Sprivel at the University of Alberta, has this kind of the research chair in symbiosis, and he published just a few years ago in Science. They found basidiomycetes, yeasts, different yeast species that were intrinsic to the lichen itself. In other words, kind of exploding the notion that lichens were simply one fungus and one algae, but that there's a complex arrangement. So David Hawksworth and Martin Grube in Germany published in 2020 a new definition. You know, they called it an ecosystem, self-sustaining ecosystem formed by the interaction of an ex-habitant fungus and an extracellular arrangement of 1 or more photobionts and an indeterminate number of other microscopic organisms. So in other words, it's a big ball of wax, and no one really understands what these other things are doing. And so there's a lot of research on that right now. You can take Rosemary Hungener in 2009, you can take a nutritional type of approach that lichens derive in a mutualistic relationship carbon from the photosynthetic partners. So it's really an amazing thing when you think of it. You have a plant kingdom, the algae, and then you have the fungal kingdom coming together in an interaction that creates a whole different world. It's like in our own bodies, right? In our cells, you know, the Lynn Margulis, right? Margulis, her thing about the exosymbionts. You know, the mitochondria in our bodies, you know, they were once free living, and the chloroplasts and leaves were once free living. So things kind of tend to get together. So maybe cooperation and mutualism is a real driving force in evolution. So it's quite, and the lichens certainly portray that. So Trevor Goward from University of British Columbia, he lives in BC, well-known Canadian lichenologist, said fungi are basically, or lichens are fungi that learn how to farm. And because of that, they can then live in very extreme environments because they don't need soil. They don't need nutrition in the soil. They just have it with them. They have their own photosynthetic partner, and they can get along quite well in very extreme environments on the surface of the planet. So that's what's quite unique about lichens. They're poikilohydric. In other words, they can't maintain their internal osmotic pressure. You know, plants will do that, right? But you get to a certain wilting point and plants will wilt. But lichens, if it's wet and rainy, they're soft and supple. If it's dry, they're dry and brittle. So they can adapt. They can adapt quite well, and they can shut down their metabolism to the point where they can go into hibernation mode and wait for the proper conditions to arrive again. So very, very adaptable to extreme environments like in polar deserts, for example, where you have a lot of lichens. And so they can live without water. This is a very common lichen in Newfoundland. Spherophorus fragilis grows on rock, and you'll see it quite often. So they can kind of shut down and hang out until the right conditions are ready again for growth. A little bit of lichen history. It's always good to know where we come from. The lichen word is a Greek word, an old Greek word, which means a rash or a wart. And Theophytus, they call him the Greek father of botany, the first one to use the term. And even today, we use the term lichen planus. I don't know if anybody has had lichen planus, but it's not contagious. It's part of an autoimmune system, and you get these warts or rashes on your system. Carl Linnaeus was the first one in his Species Plantarum in the late 1700s to put lichens in perspective with all other plants. But he didn't know what to do with them. So he just called them lichen, and he had 80 species in his Species Plantarum, lichen X, lichen Y, and so there was only one genus, according to Linnaeus. And he called them the Rustici pauperimi. In other words, the trash of the plant world. And that's how he actually described lichens. Because he was more interested in the sexual organs of plants. And that's how he, as you know, that's how he classified plants, right, according to their stamens and pistils. People thought he was a bit kind of risque. Because, you know, you can classify plants by many other means. Why use the reproductive organs? Anyway, here we are. Eric Acarius was the last student of Linnaeus. And he was really the founder, kind of the father, if you will, of lichenology, a Swede. And he took all the one genus, lichen, and then he broke it down into many other genera. So he created these genera. And this one, like a beginning to the lichens of Sweden. So he worked on the lichens of Sweden in 1798. And then he had a series of publications in the early 1800s that really opened up the whole taxonomy of lichens. He described about 800, well, not about, he described 877 species new to science. And this is on the right is Lecanora senesia, so that's the abbreviation for Acarius. He described that in 1810, and the name still holds today. So this is the name we'll use today in 2025. A German botanist, Karl Waldroth, they were looking at these algae, right, in the lichens. And they didn't know what they were. Initially, they thought they were gonidia, they're reproductive structures. So basically, that's how they understood the little green blobs in the lichen. But a Swiss botanist came along, well, he came along and said, they're not gonidia, but they're actually algae. They're actually photosynthetic partners. So this is his doctoral dissertation, the algal types of the different kind of lichen gonidia. But then along came the German Heinrich Anton de Bary, and he finally put the word mutualism and symbiosis to the relationship. He said, this is what we have. You know, lichens are actually a mutualistic relationship between an algae and a fungus. Not everybody took to this. Nylander, one of the great Finnish lichenologists, this is what he wrote. You can read it there, but he says the assertion that lichens are fungi living in a symbiosis with algae is an assertion either a pure fancy or a slander. He wanted nothing to do with this because lichens are a noble, part of a venerable group of plants, and they're nothing to do with fungi or with algae. So bug off, leave us alone, and we're fine. Actually, that kind of still holds today in some way because we have the lichenologist, you know, the journals, or Journal of Bryology, which includes lichenology as well, you know, the bryologist. So, but it took a long while, just like any kind of scientific paradigm. It takes a while for things to change and for people to accept new paradigms, new understandings. So why is Newfoundland and Labrador a haven for lichens? Well, first of all, just our geographical position. Newfoundland and Labrador is stuck out on the North Atlantic, and we go from the subarctic, Arctic latitudes right down to temperate climates in the southwest Newfoundland south coast. So we run a range of gametes, and the fact that we're on the ocean, the oceanic influence has a tremendous importance for lichens in Newfoundland. Those of you who are from St. John's, you'd recognize this. Of all the capitals in Canada, provincial and territorial, St. John's is the windiest city, the snowiest, the wettest, the foggiest. It has the most rain, freezing rain, coolest summer, and the least sunshine. If you haven't already seen that in the last few months or years. So because of that, it's perfect for lichens. The air quality is good in general in Newfoundland, quite good. I live in Sudbury. The air quality is much better than it used to be, but it's quite a... So St. John's is really important, or Newfoundland in general. And we also get this polar influence. That's really important for us. You get the cold Labrador current comes down the coast of Labrador. It hugs the coast, and all the coastal barrens of Labrador are basically subarctic conditions, even to the south part of the Labrador, and into Newfoundland as well. So you can see the sea ice, this white, you know, going into the Gulf of St. Lawrence. And notice that the south coast of Newfoundland is pretty well ice-free. That's a general phenomenon. It's quite an interesting phenomenon. But the rest of the coast, and so what can happen is that the so-called Arctic conditions come really far south. You know, I know Haley is working on that very question, you know, these disjuncts so-called. If you look at this 10-degree July isotherm, it's a dotted purple line. I mean, you probably can't see it, but you notice that this is the isotherm where the average temperature in July does not get above 10 degrees Celsius. And so you can see that isotherm comes really low. You can see here, this coastal Labrador, this northern Newfoundland, it comes really south. Whereas the isotherm is much higher latitudes in the rest of Canada and in polar Russia and Scandinavia. So in other words, again, giving Newfoundland and Labrador the possibilities for great diversity in its lichen flora. And the geology is really important. We have a lot of rock. You don't call Newfoundland the rock for nothing. And so the geology is quite diverse. The limestone barrens in the west coast of Newfoundland, that blue up the northern peninsula, Carboniferous limestone, the volcanic rocks from when the continents came together a number of times. So really important kind of substrate for many saxicillus lichens. And lichens that grow on limestone are quite different than lichens that grow on siliceous rocks. So it's, again, increasing our diversity. We have still, despite the long history of forestry in Newfoundland, forest cover is still quite strong. And we have a lot of humid coniferous forests, especially on the Avalon Peninsula. And humidity and lichens, they go together really well, especially here in Newfoundland. If you go on the Avalon Peninsula, you'll see this kind of forest, right? The balsam fir, sphagnum, feather moss forest, quite dominant. Great habitat for a lot of specialized lichens, in particular cyanolichens. And I know Riley here is doing his master's on this particular lichen on the left, Pectania plumbea, right? Plumbea, Latin for lead, so the lead-colored lichen. And they require liquid water to kind of reactivate their photosynthesis after drought. So really, they do really well in humid environments. And then the one on the right, the famous Erioderma pediculatum, it's the boreal felt lichen. I think it's endangered, or what's it classified as? Do you remember? It depends where you are, yeah. Anyway, it's not that common anyway, so it's classified in the IUCN categories. Often in Newfoundland, you cannot see the trees for the lichens, and it's really quite something, you know. Also, the different substrates, different barks of different trees have different pHs, so that can produce a different flora of lichens as well. Betula elegantiniensis, the yellow birch in Newfoundland, has a very specific lichen flora that's you won't find anywhere else in some cases. And because of our air quality, because of our kind of, you know, the conditions in the environment in Newfoundland, Osnea longissima, for example, this beautiful tinsel lichen, that can be meters long. In Europe, it's really rare. It's almost extinct in many countries. In Newfoundland, you can find it in relative abundance, on the Avalon in particular. So again, if the air quality wasn't good and if we didn't have the right humidity conditions, this species would not exist on the landscape. Well, old growth lichens, structural complexity, you know, trees of all different ages and sizes, a lot of dead standing wood, snags, and it provides a lot of substrate and habitat for lichens. What's really nice about lichens is that they're very micro-specific dependent. You know, they're not like, you look across the landscape, you have like balsam fir, you know, 100 square kilometers. But even within a square meter, you could have many different species depending on the microhabitats. And that's what's really kind of neat and unique. And like these little pin lichens, they grow only in places that are protected from rain, for example, in the crevices of snags and under tree roots. And it's really quite specialized little creatures. Again, I referred to the geology earlier. We have the ultramafic geology, you know, from the collision of the continents, so you get high base content. The Aniosquatch Mountains is a Mi'kmaq term, which means terrible rocks. They're dark, foreboding, high magnesium, basalts, gabbros, really important for a lot of specialized lichens, actually. And, okay, I'm just in front. I'm not blocking it anyway. This is the ultramafic rocks in Grossmore National Park, right? It's the mantle rock over above the crust. So there's very few lichens here, but if you take time, I was with a colleague from Norway several years ago, and we just scoured it, just walking back and forth. And we probably have one or two that are undescribed to science, you know. Just take time and to look, and these can be very important for lichen species. What's beautiful about Newfoundland and very, very important for lichen habitat is the coast. We have a lot of coast. Lots of bird droppings, lots of guano, perfect for lichens. And you get these beautiful multicolored species throughout the coast. Often lichenologists, we don't get much exercise, at least when we're there. You know, you can spend a day on this rock. You can spend a week if you really want to focus carefully. And so just, this is Mac Pitcher, one of the early great natural historians of the province, looking at a rock not too far from here actually. But you can see all the different lichens that have grown because of the bird droppings. The gulls land here and do their thing, and great habitat for lichens. The limestone barrens I alluded to earlier in the west coast of Newfoundland, really important. We've probably described 3 species new to science just from these areas. And actually this summer, there'll be a BioBlitz funded by federal money in the Great Northern Peninsula on the limestone barrens. And so I'll be there looking at lichens as much as I can. So you get a lot of calciferous lichens. And they really, the whole physiology changes. The barrens, as you know, in Newfoundland, lots of barren, lots of open heathland. It's my brother on the top of Hawk Hills. You can get polar influence here, you know, Arctic influence. So specialized flora, quite very specialized. So the heathland and the peatland in the Newfoundland, among the forests, quite significant for quite a range of habitat for these the kind of Alectoria, Sarmentosa var, vexillifera in these wind-blown areas. You'll find these lichens only in exposed wind-blown surfaces. Alectoria ocrulucra on the right, which kind of hug, and then these other lichens that grow on the rocks in these alpine areas. And then on the bogs, and you get a lot of the Cladonia species, right? clad for branch. So we have a lot of Cladonia species in Newfoundland because of our bogs and peatlands. And as I referred to earlier, the rock, Newfoundland has the rock. And because of that, because of the exposed rock in Newfoundland, it provides a wonderful substrate for a tremendous number of little black dots on rocks. So there's a lot of little black dots on rocks that look all alike, but the Porpidia genus, I'm working with some colleagues now on a phylogenetic study of Porpidia, because for a long time we were trying to figure out, identify lichens using the keys that were published, and none of it worked. You know, you could shoehorn a lichen in here or there, but it just didn't work. And so the recent phylogenetic work has shown that there's a lot of species that have not yet been described for Porpidia. So hopefully that'll be a great help for future people. So you have Porpidia, Licidia, and then Rhizocarpon. You know, we have a lot of species of these different genera in Newfoundland because of our high rock content. Some species grow only along rivers. They just want to be inundated by water. So very specialized lichen species are so-called amphibious lichens. Terra incognita is Labrador. There's been very little work in Labrador in general. On the island for the same as well. So there's a lot of possibility for potential new discoveries. This is the Saglec Fjord, the Torngat Mountains National Park. And so the Benedict Mountains around Makovik. So quite pristine, amazing landscapes that have never been examined for lichen species. Churchill River. And you get these large expanses, as you may know, right? This Picea Cladonia forest that burns every so often. And you get closer, it's Picea mariana, black spruce. And then this is 100% carpet of lichen. So that's Cladonia stellaris, the reindeer lichen, considered to be the national lichen of Canada. And this is what it looks like close up, like little starlets on top. So again, Labrador, quite something. Okay, what's in a name? Just a little bit on lichen taxonomy. Right from the early beginning, humans have always just wanted to pacify. Even for the simple reason of just for living, you know? These plants are good to eat. That's one group. These plants will make you sick, another group. These plants will kill you. You know, so the need to classify came just out of the need for survival. And so classification, naming, and just imagine when you were born and you weren't given a name, you know? You didn't have a name. You wouldn't grow. You would stunt. Certainly psychologically, if not physically, you know. So name is really important. And as we know, Linnaeus developed the binomial system in the 1700s. Before that, if you wanted to name a species, you'd have a long Latin-based description, right? The tree that grows on the edge of the cliff, and that would be the name of the species. But he developed the binomial, of course, you know, the species and the genus. This apparently is the longest species name in existence. And if you can pronounce that, great, that's good. So this is from a village in Wales. And so it's a Mycococcus, published in 2020. And that's the name they gave to the species. Thought that was pretty good. But normally we name most species based on some characteristic of the lichen or plants or animal, whatever. A Rhizocarpon geminatum, you know, Gemini, Greek for twins, the twins. So if you look this grows in Newfoundland, quite common, named by Corber in 1855, and the name still stands. As you can see on the right, it just cut into the little apothecia, and you see the two dark-colored spores in the ascus. So there's only 2 spores in each ascus. So Corber said, oh, I'm going to call it Geminatum, Gemini, the twins. So this name actually helps to describe the lichen, which is very helpful. If you like President Obama, a friend of mine, Kerry Kunson, he named it lichen after Obama back in 2009. Now, if you want to name one after President Trump, you can go ahead and do that as well. You can call it Caloplaca Trumpii or something, whatever. Another people, Jessica Allen and James Lindemer. I think you had Jessica Allen here, gave a talk there a while ago. Anyway, she and James Lindemer, they had a lichen for Dolly Parton. So they named a new species after Jepawiella Dolly Parton. Dollipartoniana in 2015. So you can name a species after anything, and anybody, you just can't name it after yourself. Latin speciere, to look at, so that's where the word species comes from. So just what we mean by a species, really it's anything that we want to decide, you know. These are common enough based on these criteria, and we'll group it in one species. You know, so we'll have different, that's kind of a real basic way of understanding. You can have a phylogenetic approach as well, Hawksworth in 2020. So just a few words now on describing some new species. I've been privileged to work with some colleagues who are specialists in different genera worldwide, and it's been really helpful and really eye-opening, I must say. So how do we classify? Well, like I said earlier, we can decide on any kind of classification we want. We can decide on body mass. So I'll take all the elephants, all the giant redwoods and sequoias, and all the giant squids, and I can put them in one genus and call them Gigantea. And that may work if the importance is body size. Obviously, it doesn't work phylogenetically. You know, now we try to classify things according to their genetic relationship through evolutionary time. You know, they share certain homologies, right? Characteristics that are passed on and shared through evolutionary time and relationships. And Charles Darwin really kind of brought this to fore. This is from his notebook on the left. This is the first phylogenetic tree or evolutionary relationship, if you will, ever put on paper, I think. So from his notebook in 1837. And the one on the right is from The Origin of Species. And the German Ernst Haeckel, you know, the ecologist, the so-called tree of life. And the word ecology came from him. He first one to use the word ecology. So when we try to name a species, we try to use as much data as we can. And morphological, chemical, kind of developmental, ecological, genetic, you know, and we try to kind of put it all together as best we can and using some more new techniques today. In the past, generally, and even today, we can describe new species and distinguish species based on morphology. For example, Ophniofarma ventosa, a saxicolous lichen on the left, and Lobaria pulmonare on the right. You'd say they're probably two different species, you know? Environmental modification is probably not that extreme, that these are actually environmental, just simply modify. So they're quite distinct. So we can do, morphology can be very helpful. But what happens when you have things like this? When you have a whole bunch of little black dots on rocks? Morphology doesn't help very well. It can. And so we can kind of go to a deeper level of the morphology and have a microscopic morphology. So we take a little bit of the, say, take an apothecia on the left, and then we'll do like a vertical cross section into the apothecium. And they have different characteristics there that can help to distinguish species. Or you can look at the spores that are in the ascus, and then the spores outside the ascus. So microscopic features are very, very helpful with lichen. And that really developed well when the microscope was invented. Before the, you know, once the microscopic technology developed, then lichen taxonomy kind of took off in many ways. As you can see, these are 4 different genera, and you can see there are 4 different spore types, quite distinct. And so the use of spore types to classify lichens has been very important through the years. So the Rhopilaspora, Rhizocarpum, Buellia, Protozaria. So you can just see how distinctly different they are. You can use the ascus as well. Characteristics of the ascus can be very helpful. Different staining possibilities, etc. Secondary metabolites. Lichens produce, I think it's what, 1,500 to 2,000 or more metabolites, you know, only in lichens. And Haley has set up, Yolanda's student has set up a TLC. I'll show a bit of it here now. That's just some of the different metabolites you can have. And Haley has set up a thin layer chromatography set up now in her lab that enables her to look at the metabolites in various lichens, and they can help the relative, well, this is all these on the bottom, all these little, oh, flit. Oh, that's good. Sorry, all these, so that's, you may have like 20 different samples here, say 20 different lichens, you know? and then you make an extract from the lichen, acetone extract, and that solubilizes the metabolites, and then you run them in different solvents on a gel, and you wait till the solvent goes up the gel, and you can see, for example, there's probably you can see the number of tracks are all the same. Say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Probably 10 of these species are the same, but then the other ones are quite distinct. So thin layer chromatography, we use in other data as well, can help to tease out the relationships among the lichens. But increasingly now, more and more, in combination with, not solely, but in combination with all the other data, we can use genetic data. And so we're basically sequencing the base pairs in the genome of the lichen. This is an example from the Porphyria study we're doing. So you get thiamine, adenine, what's like that? Thiamine, adenine, cytosine, adenine, guanine, et cetera, from one of the Porphyria samples. So using the sequences, then we can help to produce phylogenetic trees, which all the nodes refer to kind of a species or a taxon in some way. And so it can help to tease out, by taking particular markers of the genome in the lichen that are helpful, can tease out different species. So here's a one for Biotura tyrannove, which we described a few years ago. So again, genetic data, all the different kinds of data used in some kind of process, iterative process, can help. And so how to name a new species? Time. It's not always simple in many ways, because you really, especially if you look in the upper left box, discovery, the first question you got to ask, is it new? Because you may think it's new and you've never seen it before. No one in North America has seen it before, but maybe someone in Russia in the 19th century published it in an obscure journal in Russian, and so you don't know about that. And so you really got to be careful. You got to do a lot in lichen nomenclature. It's really helpful to know the literature and to historical literature. If you're going to study lichenology, I would certainly recommend to study Latin and German and Russian. And then after that, everything else is up for grabs. But it just goes to show the importance of going deep into the literature, And then you got to have some concept of the variability, because lichens can be modified by their environment in a very strong way. Lichens growing on so you got to have some sense of what the species may look like. What's the variability in the species? And then once you've kind of figured out, okay, this is new, as best we know, this is new, then you write up a diagnosis, how it's different from everything else, and you write a description. Now you can do it in English. As of 5 to 10 years ago, it had to be in Latin. But now you can do it in English or Latin. The Europeans balked at it. They said, oh, that's North America taking over again. But so you can do it in both languages. And then you publish the name. And then the name is out there, But what's interesting, has it already been published? This is a very common lichen. You'll find it on almost every rock in the siliceous, in the barrens of Newfoundland, Rhizocarpum geographicum. But this is just a few of the names that this species has had over the years. So every lichen, on average, every lichen has been named 2.5 times. So just look at the names. They were all published legitimately in a journal, et cetera, et cetera. But that's only a third of the names that Rhizocarpum has had over the several centuries. So in other words, people didn't check carefully, and you get a whole lot of list of names. In other words, a whole lot of synonyms. Here's just one example. I found this lichen. I collected it a number of times, and I have a little sample of it here. This is the white blotch. This is often what you put into the museum, into the archive. So I've collected in the St. Shots area. This is up in the Eastport Peninsula. See the white blobs on the rock face. And then again, in the Aniapsquatch Mountains, you can see the white blobs on the rock and also the major rock outcrop. And we thought, working with colleagues, we did the genetic work on it. We thought that it was a new species. And so we gave it a name, Asperlidia pruinosa, because in the apothecia they had pruina from calcium oxalate crystals. And we compared it to this Asperlidia marenii, which is a European species, but which North American workers confused all the time. So they actually named an unnamed species using an old European name, but that's another thing. But anyway, we thought we had a new species until one of the members of our team, an American who is quite versant in Saxiculus lichens, said that, oh shoot, the name was already published in 1881, but in a different genus and as a variety. So in 1881, this lichen was published as Lichenora marinii var sub adunans by Vainio, a famous Finnish lichenologist. So we had to get the type of this species. Every species has a type, a concrete example, and examine it. And sure enough, what he described in 1881 is what we called Ruinosa. So we got out of some embarrassment. We didn't name it a new species, but what we did, we named it a new combination. So in other words, we took it from the Lichenora genus and we put it in the new Aspilidia genus, and the same species name, kept Vainio's name, and then put our names, and that's it. So now it's a new combination rather than a new species. So it was kind of it was a good learning experience for us. So basically, every name that you use or produced, it's really a hypothesis, you know, and you work from that. So just a quick review on some of the lichens we've named in the last few years. Biotura terranove with Christian Princeton from Germany on trees in the West Coast. I went salmon fishing once in the Colonnade River, not too far from here. I found these lichens. I didn't know what they were. Hymenelia parva with Alan Friday in the States. So it's a new Saxiculus lichen. Cladonia ignatii with Tebo Ati and Pino Bodas from Spain. So this is found in Guelph, actually. And it occurs throughout the temperate Great Lakes, but it was a new Cladonia species. And then finally, our friend, the Aspilidia, that we did a new combination for. And there's a new one coming up now, Acroaspera minuta, with Kerry Quinson from the Czech Republic, found in the Antiopskotch and in the Port-au-Port area. That's a new Acroaspera, and it's about 300 species in this genus. So it's, again, a Saxiculus lichen. And not to blow my own horn, but Deepal already did. So there was a lichen named after me a number of years ago. And you say, you can't name it after yourself. Anyway, it's not a charismatic megafauna. It's not a panda. It's not a polar bear. It's a little smudge on a rock, but it's my smudge on a rock. So anyway. So just a quick, I'm really trying to be aware of my time here now, Deepal. Just a little bit of the history. First lichens were collected by Joseph Banks in late 1700s. And you know, he was the founder of the Royal Society of London, right? And founder of the Kew Gardens in England. I had the privilege of going to the British Museum in London and seeing Newfoundland JB, Joseph Banks, the former Arcticum, quite common in Labrador. Then to see the specimens he had collected in the late 1700s, you know, so there they are. The Moravian missionaries came over from Greenland. They were from Prussia originally, a Protestant movement. They spoke the language, and so they developed these missionary stations on the coast. The famous one being at Hebron, you know, and they collected lichens and plants and mosses and sold them to herbaria in Europe to raise money for the missions. So there you go. Lots of Newfoundland lichens in Labrador because of that work in the 1800s. Just some collection there. Nothing is more gloomy. This French Marie Bachelot de la Palais, nothing is more gloomy, he said, than these forests that are all dried out and you have all this black stuff hanging from them, these lichens hanging from the trees. And we know this quite well. You know, so that was in 1825 that he wrote that book, Voyage to Newfoundland. And a lot of work in Saint-Pierre and Miquelon. The first flora of Saint-Pierre and Miquelon came out in the late 1800s, had a lot of lichens in it. So most of those lichens are now at the museum in Paris. Yeah, another great movement. Reverend Waghorn, Church of England missionary. He didn't live very long, 49 years old, died in Jamaica after Newfoundland. And but he collected a lot of different lichens and would send them to specialists in Europe. And this Professor Arnold in Munich. And so most of the early lichen records in Newfoundland were published in the Austrian Botanical Journal in German and in Latin. in the late 1800s because of this early relationship between Waghorn and Professor Arnold. That's a letter. He lived on Monkstown Road, which is not far from here. So anyways, kind of and there was one lichen named after him, Hultin, a Swedish lichenologist, to whom Waghorn sent some specimens. He named a lichen called Pertusaria waghornii. But one of the great pioneers, really, of Newfoundland lichenology is Ted Ahti from Finland. He came here for his master's at the age of 21, looking at reindeer lichens at the request of the Newfoundland government. And he's been back and forth 9 times. He's collected 8,000 lichen specimens. He's 90 years old and still quite active. So he's an amazing man. He named one species after Labrador, Cladonia labradorica, and then Cladonia terranove because of his love for Newfoundland. And right now, the draft of his monograph for the Cladonia of the world, about 550 species, he's produced a monograph. It's in draft form now. The last one was produced in the 19th century, so this will stand the test of time. So. Okay, I'm getting right to the end. Oh yes, got 10 minutes there, Deepal. I'm almost there. Yeah. So just a bit about the future. You know, Troy McMullen at the Canadian Museum of Nature and Yolanda Wiersma here on your faculty has a number of grad students in lichenology, Niels and Haley and Riley. They're actually sitting together. Okay, good. It's a mutualistic relationship there. So there's Haley in her younger years, I think, and then Andre Arsenault from Canadian Forest Service. Also, that's Andre on the left, by the way. But so that's his dog, I think. So he's quite involved in lichenology as well. And just to show, this is the number of taxa we have so far per year for Newfoundland. And you can see it's showing no sign of leveling off. So the more you look, the more you find. And it's really quite exciting. To give you a sense of the numbers, ECFEL produced the first lichen list in 1895 for Newfoundland, 346. Right now, we're running around 1,300 on my own checklist. Just to give you an idea, Finland, which has a long history of lichenology, they're at 2,100. So this will definitely increase in the time to come. And so we're basically trying to produce an annotated catalog of the lichens in Newfoundland and Labrador and describe new species. And there's always new issues and new problems that come up. That's what I love about lichenology. You collect and you don't know what you're collecting often. And you just enter into new taxonomic and nomenclatural issues. So it just opens up a whole world of a lot of problems, quote unquote. So why study lichens? Well, just because they're beautiful and just because they exist. Why do you study what you do in your own field? You know, have you ever asked yourself the question, why do you study what you do? What is it that draws you into biology? What is it that draws you into research or whatever it may be? And so I think those are the values we have. You get to work with great people in lichenology. There's no R&D secrets. There's a generosity among men and women around the world in lichenology, and I can attest to that. Great places for lunch. And in the end, it's a real celebration of diversity, you know, beauty and joy and goodness and wildness. And really that's what I think draws us into the scientific endeavor, you know, or whatever field of study we are engaged in. Because there has to be a motivation. You don't go into science necessarily for fame and fortune, although you may, or for money, but you go into because of love and beauty and goodness, and because you're passionate about something that draws you deep. So anyway, that's the last, and I'm very thankful for your presence, and thank you very much. Yes.
Okay, thank you, Dr. McCarthy. We'll now open it up to any questions. If you are viewing online and have a question, please feel free to type it in the chat. But for now, does anybody here have any questions?
So, I got a mic. Okay.
Hey, thanks very much for the talk. So you mentioned that when you're trying to name a new lichen that you think is new to science, perhaps, you have to be looking everywhere, including the obscure Russian journal from 1885 or something.
Right.
You also mentioned that it'd be good to compare it to the type, which I'm assuming is like a physical sample that comes from a
That's
herbarium
right.
or
Yeah,
something.
each name has a type associated with it. That's
Yeah.
right, yeah.
So what's like the oldest, like some of the oldest types that you could feasibly compare it to? And are there lots of named lichens that don't have types anymore that are usable, or can you pretty much get the type for whatever you like?
Yeah, thank you. There's a number of, in the early days, they didn't have a requirement for types. So they would maybe produce a name and call it this name, but it was never typified. So the big problem, and then you get a name that's floating around in the literature or used, but we don't know what it really means because his or her description may be very scant. And so people then have to go and lectotypify. So there's a whole process of taking his or her specimens and then actually designating one as the type. So then this type, that's really important now. The type becomes the paradigm for the name. And there's a big debate now actually in conferences coming up. Can you use DNA sequences as types? Some people say yes, other people say no. You need a kind of a clearly physical type, you know. So, but we have types that go back to this, we know what they were, you know, to the late 1700s. Maybe difficult to use genetic analysis of those, but it depends maybe with the new techniques. But it's often really important, yeah, as best you can, to kind of go back to the early, like that type we ordered from Finland. It was in Turku in the museum in Finland. And so then we had to order that basically by mail and compare just physically and microscopically and chemically, you know, and that helped us then that confirmed the decision, yeah, we don't have a new species because it has already been described. But the types are really, really, really important. And now it's essential. If you're going to, each name that you publish has to have a physical type associated with it. Then they have paratypes and isotypes and those kind of things, you know. Yeah.
Thanks.
That helped. Yeah, good. Thank you.
We have a comment from Luis online saying, "Excellent seminar, John. Keep up the great work."
Thank you to that.
Does anybody else? Oh,
Yes, sir.
Thank you. My name is Cedri. I'm a new faculty here at the Department of Biology. And it's really fascinating talk. You provided us with a view of the lichen word. I think it's fascinating, really. And thank you very much for.
Thank you.
Yeah.
And you're a mycologist.
I'm a mycologist.
Okay,
well, I'm a yeast guy. Let's say.
Yeast, okay then.
It's just very, complexity of fungi is so.
That's right. Yeah.
So my question is, how would lichens differ from maybe the simplistic view of them being like a form of evolved biofilms, or and can we reconstitute them in the lab and see how they develop?
Right.
And how it would be this different from biofilm, multi-species biofilm, that dual species, the simplistic, but simple biofilm, but multi-species would be.
Right.
Do they synthesize a matrix?
As far as I know, that's been kind of the golden grail, if you will. Can we synthesize a lichen? And people have tried, and especially got it in, antiseptic conditions, and all the conditions are right, but it has never really been successful. Maybe with one species that they worked a lot on, and then you just get this kind of blob. It doesn't make any. You cannot say that that's the same thing that you find in nature. So there's so much else apparently going on that we really don't. That you can never mimic in the lab, at least so far. So anyway, it's a bit of a golden grail, and people are looking at that. Good, thank you, sir.
okay. And we will end the seminar there. Thank you very much, Dr.
Thank
McCarthy.
you. Yeah.
And thank you all for coming.
Yes, thanks very much. Thank you. Great. Well, just take the mic, huh? Yes. You can walk away. No, that's fine. Got your.